Assalamu alaikum. My name is Sayyida Hadika Noor and I belong to the Valeem's faculty. I am a teacher of biology 9700 A's and A levels, and I have experience of teaching A levels of like 4 to 5 years, and I have been teaching 5090 and 0610 biology for almost 6 to 7 years in many different places. As it is concerned with my profession, so after a teacher, I'm basically a marine biologist by profession, and I'm a research scholar in Center of Excellence in Marine Biology, and I have been doing research on molecular and genetic analysis of some marine animals. So this demo is basically a topic of 9700, and it's like from the second year, or you can even call it the second year of A level, and the name of the topic is genetic technology. So in this chapter a lot of things are there, and specifically there are some uh, there are some important principles which I'm going to tell you today, specifically the meaning of RDNA and the steps which are involved in the synthesis of RDNA, and priorily I will be telling you about the DNA. What is a DNA? DNA is basically a hereditary material, we all know it, and this hereditary material contains all the informations from our parents to offspring. We all know about it, right? Now, the most important part is that after studying DNA so well, scientists have evolved themselves into another conclusion that we can even edit the DNA, which are the basic codons present in our body. They are editable, and scientists have been doing experiments for that, and they call this field genetic technology. The aim of genetic engineering is to remove a gene from one organism and to transfer it to another one. So there will be a third category or third type of gene, and they call this technology either gene engineering, gene editing, or gene technology. Now, after so many experiments and researches, we can even easily find it from the internet. Scientists have come to this conclusion that it is very, very beneficial and lucrative for the world of humans, as by editing the desired gene, we can even edit the plants, we can even edit the crops we are eating, we can even edit the food animals we're consuming in our daily life. So it is a very different type of technique, and in Pakistan there are a lot of centers which are working on it, specifically Center of Excellence in Molecular Biology in Lahore. It's known as CMB, and this is a very interesting technique which we are just going to discuss about. So in this technique there are a lot of steps involved, and the end product of that technique is the RDNA. RDNA is like DNA, we know that it's deoxyribose nucleic acid, but what is R? R is the recombinant one. Like, it has got the combination of two different DNAs, and it's like redesigned by the humans. It is like changed, modified by the humans. So the organisms which are basically formed from it are genetically modified, and we can even call them GMOs, or genetically modified organisms, and the DNA they contain is known as the RDNA, or the recombinant DNA. As we all know about the basic structure of DNA, that it is a composition of three basic materials or molecules. In biochemistry, we can say the first is the phosphate group, the second is the sugar, which is basically the deoxyribose one, and the third one is the nitrogenous bases. And there is a variety of nitrogenous bases in the DNA, like it could be guanine, it could be cytosine, it could be thymine, or even adenine. Now, the genetic code are universal. It has got a different identity specifically when I talk about a species to species, and they contain the information even which are inside us, and this is going to be expressed phenotypically in any organism. They contain the secret codes. Now, these codes are basically decoded by the amino acids which they form in the form of either enzymes or any hormones. And this is really important information which is hidden in our DNAs, and you can even say that it is very important even for a lot of metabolic reactions which are going to happen in our body. Plus, there is this different, you can say, information of different organisms even for their identifications. So these codons can be used for the identification of specific species types as well. Number two, these informations can be edited by the required or by the choice of any person, and after editing it, the desired information can be easily recombined from two different types of DNA. So when we talk about the combination, scientists have been trying to, artificially edit the genes, and they try to change the genes. They call it RDNA, and the species which are, can be modified from it are known as the genetically modified organism. Even they are sometimes called as the transgenic organism. Like, the genetic information has been transferred from the one organism and transferred taken from one organism and transferred to another organism. So these are basically the concept of GMOs or transgenic organisms. So if it comes about the exact definition of the technique, then what is it? So it could be defined as that it is a technique used to, deliberately modify a specific characteristic of an organism, and this technique involves the removal of a gene from one organism and the insertion of a gene to the desired organism, and this whole scenario and process is known as the genetic engineering. In this process, a lot of steps are involved. First of all, scientists have to decide that what type of organisms they want, number one. Number two, after deciding, they need to identify the specific gene that, okay, this gene has to be taken out and this is our desired gene. As I have just explained it, it's not that easy process. Scientists need to, you can say, visit the whole genome of an organism if he is going to change a small piece of it. So it's a very different, or you can say it's a very time-taking process, but it can be done for the benefits of mankind. Step number two, the isolation of the desired gene. Isolation of the desired gene is done by cutting the chromosome part by using an enzyme, and the enzyme is known as the restriction enzyme. Using reverse transcriptase enzyme to make a single strand of complementary DNA from mRNA. As we already know that RNAs are used to make the process or the copy of another DNA. So here cDNA can be easily made from the mRNA. The third one, creating the gene artificially using nucleotides. After having a cDNA, the complementary strand can easily be created. The fourth one, multiplication of gene by a reaction known as PCR. PCR is a very different type of reaction. In science, as we call it, polymerase chain reaction. Poly means many, and mer means molecules, and ase is used for the as a suffix for an enzyme. So basically, polymerase means that there are a lot of molecules of enzymes used in this reaction, and after passing these sequences of events, we can have copies of, or you can say, a lot of multiplication clones of the genes which we have just created from the nucleotides. The next step is to transfer the correct gene from the PCR results into the organism, and we call that organism a vector. And the last stage is the identification of the cell with the new gene by using a marker, either it is cloned or not correctly. Markers are like standards. They're normally used by the scientist for at so many stages just to identify the already marked or corrected gene, just to compare that is it the correct gene which we have cloned correct, or does it match with the previous information or not. Genetic engineers need the following to modify an organism. They need the enzymes, and enzymes could be restriction endonucleases, ligases, reverse transcriptases, some vectors. Vectors are basically, they are used to deliver genes into a cell like plasmids, viruses, and liposomes. Markers are basically the codes. Okay, so basically markers are like the genetic codes. They are used to identify the organisms by tracking them inside any organism. There are a variety of markers which are used in the world. For example, GFP, green fluorescent protein, which is used under a UV lights, or GUS, glucuronidase enzyme, which transforms colorless or non-fluorescent substrates into products that are colored or fluorescent, right? So it is a visual representation of the first step, that is identification and isolation. As we can easily see from here that in the first step, the identification of the desired gene, for example, insulin, is being done from the human pancreas. Number two, isolation of the desired gene is done, and the gene of the pancreas is being taken from the animal cell, and the gene which was taken from the animal cell was basically like a single strand of the basic information. The basic information was taken from the pancreas, and with the help of free nucleotide, it can be easily produced as a cDNA. cDNA is basically complementary DNA. Complementary DNA was produced from the mRNA. mRNA was the information which we got from the insulin produced from the beta cells of the pancreas. Now, if we look at the gene or the DNA which we have after cDNA and mRNA, which we just have, isolated, so it has got two ends. These ends are halfly completed. They are missing. There is a part which is missing over there. So the part which is missing over there is basically known as the sticky ends. These sticky ends are used in the third step. These sticky ends will be attached by the vector, but before it, we need to make a lot of copies of them. And this is done with the help of the PCR. PCR is a polymerase chain reaction. In this reaction, we can make a lot of copies of the desired gene. When a lot of copies of the desired gene is in the results of PCR, we can easily transfer the desired gene by a vector. A vector could be like a plasmid inside a bacterial cell, and then we can see that how this recombinant plasmid will behave. And it can be easily identified by a gene marker as well. And now this bacteria is the transgenic organism, because it has a transfer gene, or you can say a recombinant DNA in itself. Okay, so basically in cloning, cells with the desired gene are identified by the markers, and the copies are being made from the single organism which was modified, and the making of the copies which are exactly similar to the parent one is known as cloning. Alright, so in this lecture we learned today about three things, and this is the summary on the screen. Number one, the recombinant DNA is formed with the combination of two different DNAs. Number two, GMOs or genetically modified organisms are those organisms which are reproduced from the recombinant DNAs. Number third, the basic steps which are involved in the formation of transgenic animals and plants or GMOs are identification and isolation. Number two, the transfer. Number three, polymerase chain reaction. And the last one is cloning. Okay, so this was all about my lecture today, which was related to genetic technology and GMOs and transgenic animals and plants. So I'm just signing off from here. I'm Sayeda Kanur, a teacher at Valeem, and it is a very good platform for you guys if you choose it, especially in online learning. I'm always here for you guys. I'm always here for help. Thank you. Allah Hafiz.